Welcome to Akhand Vidyashram, the first institute of impeccable wisdom. Divyank, the perfect human DNA model. My mission, the impeccable wisdom, knowing which everything is fully known. My greatest discovery is Divyank, the divine design of nature with which the perfect objects are designed and created. Now we have come to a very special state of evolution. Now we are going to see how Divyank, after being recognized, conceived, and revealed, can be applied in day-to-day life to understand perfect human anatomy, physiology, biochemistry, psychology, and thereby we can prevent all the health issues, all sufferings, all the problems. We can curtail aging as well as reverse aging. And so that we can be healthy, wealthy, wise, and happy, and the whole human society. Now only we are coming to the applied part, and I'm sure things will start becoming easier and easier hereafter because you'll be able to apply into our own life and make a difference to ourselves, our society, and human race by and large, and the world peace on the whole. So let's start with the applied part, which is very important discovery when come to the question of spiritual creation or spiritual evolution, or the human being's evolution from whatever. So first thing which comes to my mind is always genes are inheritance are the model with which our human beings are designed with. As I told in the beginning, human beings are the greatest microcosmic representation of the macrocosmic universe. As we see the entire universe, the way it is designed, that's what ancient Indian sages and the Western philosophers and prophets also have mentioned, that we are made in the image of God, what prophet Moses talks about it, and I'm sure almost every religion talks about it, that we are made in the best design of the world. And Indian scriptures also talk that we are also created as a low pollen, or the guardians of the world, or custodians of the world. If we are created in the model of divine consciousness or the God or nature by and large, and these are represent, and I think one model which comes to the mind is our genetic structure, our DNA structure. It has to be on par with universal consciousness. From that point of view, spiral galaxy, if you take the image of spiral galaxy, the length and breadth from golden rectangle to golden Divyank rectangle to Divyank, I think the picture is very clear. Now let us see whether that picture we can see in our genetic structure itself, with which we are further evolved. So the first major step towards finding that genetic structure, credit goes to James Watson and Francis Crick and their Nobel Prize of 1953. And now let's understand that particular genetic structure which they reveal to the world. And their model is like this: diameter 20 angstrom. One complete helix is 34 angstrom. Minor groove is 12 angstrom. Major groove is 22 angstrom. Then you have sugar phosphate backbone, and then you have a nitrogen base pair, and number of strands 10, and distance between each strand is 3.4 angstrom. This is the essence of the model which they created, for which they got a Nobel Prize, and both sides models have been made. And subsequently the cross section discovered by other people about the B-Double Helix has also been placed. Now I'm sure from this point of view you're quite clear about the shape, size and structure of human B-double helix, which is specific to human beings. And then at this juncture also I tell there are 22 pairs of autochromosomes and one sex pair. Is this 20 pairs of autochromosome is also accidental or coincidental, or it is also part of divine because it belongs to, again, Divyank 22 by 21. Anyway, we'll not go into detail at this particular juncture. We'll see that later. So thanks to Nobel Prize, thanks to the work of James Watson and Crick, that we have a model to build on. Now, this particular picture I have is very well known in the internet, in all the academic circles internationally. It's taken from internet, and both the things it's a joint venture by James Watson, Francis Crick, Rosalind Franklin, and Maurice Wilkins. The four of them have started the work, and two of them got Nobel Prize. Two did not come, but I'll not go into the controversy why these kind of things happen. What goes into it, I have absolutely no idea what happened. Not get into that, but there is one point which I would like to make at this particular juncture. I, as a spiritual scientist, have been blamed by every other research scholar and other people who are connected with research directors to coordinators to everybody, that it's not easy to make a team with me, or they say I am not able to make a team of research scholars and make impact. No, I do agree with all the humility. My 30 years research, which is unique, unusual, path-breaking, even after 30 years, nobody has been able to replicate that particular work, is unpublished. The reason being unpublished is not that I didn't want to publish, but nobody was prepared to publish it in the original form as I have conceived. Everybody was trying to tinker with it and give it a different color, which would have been very different from what I had conceived, what I have experienced, what I was trying to explain. That is one side which I would like to put it across for the world to know why there is nothing published in my name in any of the journals or any other thing where references are involved, where people I have to go through the process of being, part of the team to make, this is how it has to be presented. It's not to be the original work, which I was not prepared to do. But it's not that I never wanted to make a team. As a matter of fact, my first team goes back to school. Arun Kumar, I made as a team, I brought him. He got the second position. But what happened thereafter? All these people who I brought, make them understand, they get into ego ki all they have done is only by themselves. And at particular infancy stage itself, they find this is a great work and they hijack it before the coming. And this is what I have seen every 2 years. I find somebody I'm preparing, they hijack that. But I'm glad that they hijack that, and I start from there to the next level. I forget about that particular, let that particular person take. Now I can go to the next. This going next. I come to this particular stage. I'm happy. I could not make a team because I could not compromise. Because even when you go to Sri Mata West University as an editor of the university and chairman, it had to be one-man show. And everyone says, you want to be in a driving seat, you will never be able to make a team, but I'm glad. I want to be driving seat, I want to be perfect, I want to bring with impeccable wisdom, I cannot compromise. Since that strength of non-compromising nature, non-aligned, and bringing, I think, has brought me to Divyank on my own with the help of very decent, civilized research scholars and good human beings who helped me to uncode or decode certain things at particular stage. For example, Professor A. Namachwam was great help for me for my PhD work. Dr. A.C. Gangadhar was great help for converting analog digital into digital for power spectrum analysis. Subsequently, in Chettinad, Professor Govindraju was very helpful to sit with me, with patiently letting me find the last 5 digits of Divyank, and things are done. It's not that I have done, but unfortunately, they could not be made into full team. I can acknowledge that their contribution to that an extent, and that was the rule, and I think let's leave it at that. So I do believe that many people play a role. For that matter, I wouldn't have been able to make Divyank as today had it not been the ancient Indian sages, what they had written in Vedas and Upanishads, and what modern scientists and mathematicians have done the world around, the kind of equipments we have, the kind of methodology we have, kind of softwares we have, which is contribution to a different thing. So indirectly, I have to say it is not my single effort. Without all that, I wouldn't have been able to do. But somebody had to take full charge to make sure the final product is impeccable wisdom. This is what I'm glad ki my being very stubborn, no compromises on impeccable wisdom, has brought me to this level. Now, from this particular concept, let me start the journey of analyzing great works and saying, good, very good, but not as perfect as Divyank demands. So, let's first unfolding come with DNA model itself. I've given that. Now, this on top there are two cross sections of B-double helix, and as I've been saying from the day one, I want to convert all mathematical designs into symbolic representation and create a new language of symbolic yantra vidya or akhand yantra, etc., etc. So that you see the symbol, you should immediately know. Other two symbols, you find the same reflection is there. You find right in the middle, outside, you have 22 by 21 and 1 dot, which is equal to 1 by 21 dot. Then in second part is the 10 pentagons, which represent power of 10. Inside you have 5 structures which represent 5 digits. So same Divyank 22 by 21 outside, power of 10, 10 pentagons, and 5 digits inside, equivalent to that. Now see the same structure and you see the resemblance in both the structure which others have created independent of me. Am I trying to acknowledge them that they've also done independent work and we are doing independently, but trying to look for things. Maybe at the same time will come when we'll all merge into one, and with from impeccable wisdom, we'll try to rectify other deviations are there in other work so that we have a perfect model. And this is the objective of this particular work too. Point of view, fair enough. As I said, phosphate sugar backbone on either side is a part of B-double helix or DNA structure. And then you have nitrogen base, as you can see down, which have 2 pairs of nitrogen bases, 1 and 2. Now let's see this picture a little more in detail. The one which I have created as far as divine chemistry nucleic acid is concerned. Left side is the backbone, starts with the outer phosphate group and inner deoxyribose sugar. Now, as you see in the middle, one side you have 5 carbon end, other side you have 3 carbon end. If the top is 5, the bottom will be 3. If top is 3 end on the right side, bottom is 5 end. This 3 and 5, is that coincidence? No, they are part of Divyank ratio. Second thing, in the central you have nucleotides. Now you have one pair, adenine plus thymine. This pair has got 2 hydrogen carbons. There is another pair, guanine and cytosine, which has got 3 hydrogen bonds. So 4 nucleotides, and the combination is there, is very, very clear, you can see. Now 3 hydrogen and 2 hydrogen is also in Divyank ratio. Now I'm sure nucleic acid, this particular structure as such, is now fairly understood. Now let's say, can we reveal something, can we find something much more interesting, mathematically designed, which brings down the divine nature, how beautifully it does. And this is also a structure where I have also discussed with one of my students. We were trying to unfold, and in the process we did a lot of work, and she was MSc Keetnavi. Now what we worked together, what was like this. The model created by Shibhushan Sharma again, since it's based on Divyank, which has been done over a period independently by me, with the help of some people at different stages single-handedly. I agree. But what is the model which I have created which says diameter is 21 angstrom, not 20 as Watson and Crick says. They say there are 10 spirals, and I said there are 10 plus 3, 4, 4 spirals, and each spiral is 22 angstrom, and they call it major groove is 22 and minor is 12. And major groove is what I'm talking about as a 22. So he also says, they also say 22, I also say 21. They say diameter 20, I say 21. Full length is 33.9854 angstrom, which is equivalent to 34. And incidentally, they also talk of 34 angstrom. So there is not much of a difference on that. But there is something much bigger appeared as I was trying to explain it to her. Let's explore. There is something deeper which nobody has even looked at it. So I looked at the phosphoric acids protons, hydrogen 3, phosphor 2, and oxygen 4, equal to 50 protons. Deoxyribose had 72 protons. Then nitrogen base pair had a completely amazing kind of proton structure which surprised me and of course her too. First pair of adenine had and thymine had 136. Adenine has 70 protons, thymine has 66 protons. Put together they are 136 protons. The second pair of cytosine and guanine also had 136 protons. Cytosine had 78 and guanine had 58. Look at that thing, 70 plus 66. Here you have 78 plus 58. The difference is 16. One only difference is oxygen. One has been removed, another has been added. But whichever way, the number of protons kept alive, same thing, which was surprising, amazing, and I thought, oh my God, nature can play up to that an extent, and I'm sure there are many more secrets in this particular Divyank DNA, which discuss slowly for the time being at this juncture to say the model by Watson and Crick was great start. Now we have taken it to one particular level, and I'm sure subsequent generation of research scholars could find even much more deeper things and add value and then go on to make it better and better. And that is what science is, that is what evolution is all about. And hope and wish you all. At this juncture, I can only say thank you very much. It has been wonderful sharing this particular information which we have discussed over a period, and I'm happy now Divyank has started finding its application in anatomy itself, and nothing better than genetic structure from which the entire thing is going to be developed hereafter. Okay, thank you very much.